Chapter 14, the Brain and Cranial Nerves. So the purpose of this chapter is going to help us understand how the brain is organized, how we protect it, and how we nourish it when we supply it with blood. We're going to compare and contrast the various areas of the brain and the functions associated with those areas. We're going to discuss the functional organization of the brain, and we're going to end this lecture discussing the cranial nerve structure, location, and function. So we're going to start by outlining the principal parts of the brain. We'll start with the largest portion, and again, we're looking at a mid-sagittal cut. We start here with the cerebrum. Again, this largest portion, we could outline this largest portion of the brain. The next part we're going to look at is the diencephalon. So we'll put cerebrum number 1, diencephalon number 2. The diencephalon includes the epithalamus, the hypothalamus, and the thalamus. So we outline these regions as the hypothalamus, all directional. So the thalamus is egg-like shaped structure in the center of the brain, thalamus. Below it, hypothalamus, and then continuous with the pineal gland, this little region up here is the epithalamus. Now, we go on to the brainstem, and the brainstem is going to include the midbrain, the pons, and the medulla oblongata. We could circle the midbrain, pons for bridge, and the medulla oblongata. And then we look at the tree of life. We look at the cerebellum. This is this second portion of our brain right here. That is our cerebellum. And then lastly, moving through the foramen magnum and then through the vertebral foramen down the spinal cord is our fifth portion, the spinal cord. The next step is to talk about protection. When we did the spinal cord in our last lecture, we talked about the meninges and how we protected that delicate structure with bone and connective tissue and fluid. Well, here there is no difference. Well, a little bit of difference. We look at the similar structures. One, we protect with bone. Those cranial bones, they're sutured, making this encased cavity, encasement of bone, big protection. Then we get the connective tissue, the cranial meninges. Now, a little bit different from spinal meninges. The layers are the same, but the spaces are different. So we have the pia mater, the arachnoid mater, and the dura mater. And again, if we go from superficial to deep, the pia mater is what is physically fused onto the brain matter, the cortex of the cerebrum. Now, the cranial dura mater is composed of 2 layers. So this is a little bit different from spinal cord. The spinal cord did not have these 2 layers. It had an epidural space, whereas in the brain, surrounding the brain, we do not have that epidural space. But the 2 layers are the periosteal layer, so the layer that's pretty much touching those cranial bones, and the meningeal layer, the layer that's making up and touching the meninges. Now, from there, we have the arachnoid mater, and again, the same subarachnoid space where CSF is going to flow, cerebrospinal fluid, and then the pia mater touching to the actual brain. Now, lastly, the last level of protection is the cerebrospinal fluid. Now, it's going to flow just like the spinal cord. It went through the central canal and around the spinal cord. This is going to go into the ventricles of the brain, so within the brain, and then out and around through the subarachnoid space. Perfect. Let's zoom into the cranial bones and the meninges. Now, there's a couple interesting things we see here. So let's start with our skin. You can see the layer of fat. We have our parietal bone, and then the dura mater. And again, you can see the 2 layers, the periosteal layer, the meningeal layer there, and they're named here, 1 and 2. Then we have something weird. Look at the subarachnoid space. So it looks just like we saw with the, we'll zoom on in a little bit. Looks exactly like we saw on the spinal cord, keeping that space open, those extensions of the arachnoid mater. And then here's our weird things. Look at these little mushroom-like structures. Okay, these are called arachnoid villi. Now, notice they move into this superior sagittal sinus. So this is venous blood flow. So the question is, what are these? What's the function of these? What do they do? Well, the answer is pretty interesting. So now, to explain this, we go back to the ventricles of the brain. We go back to those neuroglia, those ependymal cells that are making CSF within the hollow spaces, the ventricles of the brain. So we're making about 600 to 700 milliliters of CSF a day. We're only circulating about 100 to 150 milliliters at any given time throughout the day. So the question is, as we turn this amount of CSF over about 3 to 4 times a day, how do we recycle it? Where does it go? Well, the answer is and are these arachnoid villi. So what happens is CSF, as more CSF is created, the CSF moves into these little pouches, into these arachnoid villi. And as the pressure builds, they act as one-way valves and they'll squirt that excess CSF back into the superior sagittal sinus where it could go and get recycled throughout the rest of the body. So that's a pretty interesting note. And again, we'll talk about this in a little bit. Now, other extensions, and we'll look here. Again, it's hard to see. I'll draw in the green. Of the dura mater are going to be our seat belts of the brain. Now, we're going to go through the 3 seatbelts of the brain, but here's one of them, the falx cerebri. So again, remember that as we talk about how we stabilize the brain. Now, as we keep on moving in, we have our pia mater, then you can see it's right here in red, fused right to the cerebral cortex, and then the arachnoid mater and here in purple that gives us those subarachnoid or those arachnoid villi, and then again the extensions to open up that subarachnoid space. Now we lead into the seat belts of the brain. So these are extensions of the dura mater. Now we already found the falx cerebri, so here's one. Now notice it's going to go between the left and right hemispheres of the cerebrum. So it is this banding structure here of connective tissue. Notice suspended within it is the inferior sagittal sinus here, and that superior sagittal sinus that we just saw where those arachnoid villi move into to squish those excess CSF in. Now, the next falx, so again, this goes in a longitudinal type of plane, is the falx cerebelli. So look at falx cerebri, separates the left and right hemispheres of the cerebrum. The falx cerebelli here separates the left and right hemispheres of the cerebellum. Now, the next extension is a little hard to see, and the reason why it's hard to see is because of the cut we're in. This is a sagittal cut. If we were to make a almost like an oblique cut, or at least a transverse cut, we'd be able to see our third seatbelt, the tentorium cerebelli, and it lies right here. And the reason why we can't see this is because it works on a level like a plane like this. And what it does, if we were to look down into, so look down into, this is a transverse cut, look down into the skull, we see the falx cerebri would look like a line like this. The falx cerebelli would look like another line like that. And then spanning from left to right would be this tentorium cerebelli. And what this connective tissue does is kind of isolates, separates, and stabilizes the cerebrum from the cerebellum. Blood flow to the brain. Blood flows to the brain through the vertebral and carotid arteries, flows back to the heart through the jugular vein. So, and when we do blood vessels in AP2, we'll go through these, but just realize that the major arteries coming from the heart delivering nutrient-rich and oxygen-rich blood to the brain are our right and left common carotid arteries, and they do come off of the aorta in one way, shape, form, or another. The right common carotid is a little bit unique because it comes off of a trunk known as the brachiocephalic trunk. The left common carotid has its own direct shunt off of the aorta. Now, we break up our carotids into external and internal, and they're pretty straightforward because they name where they go. The internal goes deep into the brain, whereas the external feeds the outer portion of the face, the nose, the skull, the cranium, the skin, etc. And then in back here, going through those foramen, those transverse foramen, in the vertebra, we have our vertebral arteries. Now, as once blood is used and abused through the brain and then the face and the skin and the tissue, it works through our capillary beds and then reverses its way down via our venous flow. And then again, moving down the internal and external jugulars, and we work our way down back into the superior vena cava where it re-enters the heart and the right atrium. Now, blood flow is super important to the brain, and we see that the brain utilizes close to 20% of the body's oxygen supply. So if you look at weight to size to compare to other organs in our body, right, our brain takes about 20%. That's a massive amount of oxygen. And then when this oxygen and nutrients are interrupted, we know that we can sustain injury. Now, we can get a weakening of our body, this tiredness, this mental confusion. We get permanent damage, right? So if you sustain permanent oxygen loss, whether it be from being submerged underwater too long or in a worst case, horrifying scenario of somebody strangling you, the brain will shut down, you can pass out, and then permanent damage can ensue. And then again, if oxygen is interrupted for a long period of time, we get death of all the brain cells. And again, these brain cells cannot regrow, so that is permanent. Now, we talk about oxygen, we talk about nutrients. So we deal with a very hungry type of tissue. So we say that the brain takes 20% of oxygen. Let's look at glucose. The brain requires, at rest, 60% of available sugar in our bodies. That's a massive amount. We don't realize how much energy our brain really needs to run appropriately and properly. Now, what happens if you have glucose deficiency? Say you're starving yourself, or you're fasting, or you're on a weird diet that removes carbs or things like that. Well, your body and your brain specifically can suffer. Now, what happens is you may produce mental confusion, so you are confused about what's going on around you. You could have dizziness, which can lead to somebody passing out and causing head trauma or other injury from the fall, convulsions, and ultimately, in worst case scenario, unconsciousness. Another important protective border to our brain is the blood-brain barrier. It's formed by tight junctions between capillary endothelial cells and astrocytes, which secrete chemicals which maintain the tight junctions. Now, it is a highly selective, semi-permeable border that prevents solutes in the circulating blood from non-selectively crossing into the extracellular fluid of the CNS, or central nervous system, and this is where those neurons reside. The blood-brain barrier is going to allow the passage of some molecules by passive diffusion, as well as the selective transport of various nutrients, ions, organic anions, and macromolecules such as glucose, water, and amino acids that are crucial to neural function. The blood-brain barrier restricts the passage of pathogens, the diffusion of solutes in the blood, and large or hydrophilic molecules into the CSF, while allowing the diffusion of hydrophobic molecules like oxygen, carbon dioxide, and hormones, and some small polar molecules. The cells of the barrier actively transport metabolic products such as glucose, and this is going to move across the barrier using specific transport proteins. Now, from a clinical perspective, the blood-brain barrier acts effectively to protect the brain from circulating pathogens. Again, remember, pathogens are things that want to do us harm, virus, bacteria, protozoans, etc. Now, and accordingly, blood-borne infections of the brain like prions are relatively rare. Infections of the brain that do occur are often hard to treat because it's hard to get into this area because of the blood-brain barrier. Antibodies are too large to cross the blood-brain barrier, and only certain antibiotics are able to pass. So in some cases, these drugs have to be administered directly into the cerebrospinal fluid, where it can enter the brain by crossing the blood-cerebrospinal fluid barrier. Let's take a look at cerebrospinal fluid, or CSF. Now, we know that CSF is a liquid. Its function is to protect the brain and the spinal cord by flowing in the brain, in the spinal cord, as well as around it in that space of the subarachnoid, so the subarachnoid space. Now, it's also going to function to protect. So we talk about protecting against chemicals as well as physical injuries, and it's going to carry oxygen, glucose, and other important substances from the blood to the nervous tissue. Now, we can outline the three main functions. So we have this mechanical protection. So again, we're protecting against physical injury. It's the flowing around and in the brain, around the spinal cord and in the spinal cord that's going to act as that cushion. Homeostatic function, maintaining stability. Now that stability is going to be circulation, giving nutrients, oxygen, glucose, and other important substances. And then lastly, again, circulation, circulating roughly 150 CSF, 150 mils of CSF at any given time, that will recirculate and then replenish about 3 to 3.5 times a day. So we're going to revisit those ventricles, those hollow spaces of the brain, and the choroid plexus. So let's think back to those ependymal cells and those neuroglia that were lining the hollow spaces of the brain, lining those ventricles, and see exactly what they do. So remember, the ventricles are, again, the hollow spaces, and they're going to contain the cerebrospinal fluid. Now, those neuroglia, those ependymal cells that are lining these hollow spaces, are making cerebral spinal fluid. So again, remember those ependymal cells, and you can see them right here. And we know that the function of these guys is going to be to maintain the volume of CSF. And again, if you recall from our previous lecture, we're circulating anywhere from 80 to 150 milliliters of cerebrospinal fluid in and around the brain and spinal cord at any given point during the day, and we are recycling it. So here's where we're making more every day, and we recycle it about 3 to 3.5 times a day. So let's go ahead and trace the flow of cerebral spinal fluid through and around the brain and the spinal cord. So we can really pick any one of our ventricles. So remember, we have the lateral ventricles. Now, there are no first and second ventricles. Those largest ventricles that occupy the majority of the left and right hemispheres of the cerebrum are the lateral ventricles. So let's start there. So we start right here. We'll start at the top, and we'll say that the choroid plexuses, so those ependymal cells that line, and you can see in the lateral ventricles, so if we zoom in right here, we have those ependymal cells that's making CSF, and you see the purple arrows moving through and into that space. Now, we do have a little membrane that we'll talk about when we look again at the brain, especially in lab, that occupies the space between the left and right lateral ventricles. And it, again, if we zoom on in, it'll encompass this area right here, and it is called the septum pellucidum. When we do the brain dissection in lab, you'll physically pop that septum, that little thin membrane, to get to those lateral ventricles. So let's go back to our flow. So we start again at the lateral ventricle. The choroid plexus is making CSF. It is going to the lateral ventricles, down through the interventricular foramina. So again, remember, foramina is plural for these holes, into the third ventricle. From the third ventricle, it passes through the cerebral aqueduct that lies within the midbrain, down into the fourth ventricle through the foramen of Luschka and the foramen of Monroe. These are also known as our lateral and median apertures. Then to the subarachnoid space, and we're now out of the brain into the spinal cord, into the arachnoid villi of the dural venous sinuses. So again, remember those little one-way valves, those little tufts, the extensions of the dura mater that extend into the venous sinuses, are going to squirt that used and abused CSF back into circulation. Now, once it's in that venous blood, it goes back to the heart and lungs, where it's going to get reoxygenated and then can go one of those three passageways again. So the same thing, whether it hits the third ventricle first or the fourth ventricle, it really doesn't make a difference, because we follow the same pathway down and back to the heart. Now, you have a little hyperlink here. This is an animation for the brain. It runs through an overview of the nervous system, and you can click on this through your slide sorter. Now, again, remember, you must be connected to the internet in order to run this system, run this slideshow, and you must be in slideshow mode in order to see this animation. Let's look at some of the components of the brain. We're going to start with the brainstem. So the brainstem is composed of three parts. First, the medulla oblongata, and we see that right here. Okay, it is this bulbous portion right here. We're then going to have the pons. The pons would be like our bridge. So this region here that bridges the medulla oblongata to the midbrain. And we see the midbrain right here, which includes the stalks, the feet that kind of anchor it in, which is the cerebral peduncles. We're going to look a little more closely at the medulla oblongata. It is continuous with the superior aspect of the spinal cord and contains portions of both motor and sensory tracts. Now, we can take a look at this picture, and we see a couple things terminology that we're going to introduce and discuss. First is going to be where these tracks cross. So we see these crossing of these tracks here. Anytime tracks cross, these happen to be the pyramidal tracks. The word we're going to use is decussation. So right here, crossing is going to be decussate or decussation. So again, we see that we have the right part of the brain controlling the left part of the body, and vice versa. The left part of the brain will control the right part of the body. That's where we get this crossing over. Now we start down here inferior, we get the spinal cord, and then we have other tracks. So we have another track called here the anterior corticospinal tract, and we have this track here, the lateral cortical spinal tract, so anterior versus lateral. Now again, remember these are spinal, not spino. So because of the spinal, these are motor tracks. Let's further examine all the different functions and parts of the medulla oblongata. So we go back to those pyramids, those pyramidal tracks. Again, remember these are motor tracks, motor fibers from the brain to the medulla oblongata. The second function is going to be our vital reflex center. So we're talking about the oldest part of our brain, these cardiovascular and respiratory centers that are going to maintain the rhythmicity of the heart, the rhythmicity of breathing, allowing for blood to flow without you having to think about it. Next are our non-vital reflex centers. These would be things like if you were to touch a hot stove, again, those flexor reflexes, cross extensor reflexes, etc. Again, not vital for life function, but again, they are reflexes that help protect us. Number 4 are our inferior alveoli nuclei. They serve an important function for motor learning and function. Next is our gracilis nuclei or gracilla. This is the lower nuclei. Their job is to deal with fine touch and proprioception, which is balance of our lower body. So if we have something for our lower body, we have something for our upper body, and that's what the cuneate nuclei are for. Again, same thing, fine touch and proprioception or balance of the upper body. Now the two nuclei are going to help form another tract called the medial lemniscus. Number 7 is our gustatory nuclei, and as its name implies, this is for taste. This is what governs our sense of taste. Our vestibular nuclei is going to be responsible for our sense of balance in our ear. So the medulla oblongata runs our ability to maintain balance, be it static or dynamic equilibrium. The last, number 9 nuclei, is going to deal with a series of cranial nerves 8, 9, 10, 11, 12. Now these 5 cranial nerves that are governed through the medulla oblongata is our cochlear branch. So this is our actual one branch of the vestibular cochlear nerve that deals with actual hearing. Our glossopharyngeal nerve, which deals with the back of the tongue and the pharynx for things like swallowing. The most important cranial nerve here, cranial nerve number 10, would deal with pretty much all parts of our body. It's one of the most important cranial nerves that we'll talk about, and it'll come back again to revisit the function of this cranial nerve when we do AMP2, specifically our autonomic nervous system. Cardiovascular, again and again, this nerve touches all our visceral organs. We have the accessory nerve, and lastly, our hypoglossal nerve. So again, these 5 cranial nerves have their nuclei originating in the medulla oblongata. The next part of the brain we're going to look at is the bridge, the pons. So it is, again, this rounded bulbous portion linking the brainstem to the medulla oblongata here. So the pons is located superior to the medulla oblongata, and again links part of the brain with another by way of tracks. So the pons connects part of the brain. Again, there's our word, it's a bridge. It's going to be the origin for nuclei of cranial nerves 5, 6, 7, and 8. These nerves include the trigeminal nerve, the abducens, facial, and vestibular branch of the vestibular cochlear nerve. Remember, the medulla oblongata dealt with the cochlear branch, so we have 2 branches of this nerve, and we'll just write in that vestibular and cochlear branch. The pontine nuclei are going to relay nerve impulses related to voluntary skeletal muscle movements from the cerebrum to the cerebellum. The last function are these pneumataxic and apnoistic centers. These are control centers for respiration. So the pneumataxic center is going to really deal with rhythmicity, right? So rhythm, keeping that balance. The second part is going to be the apnoistic center, and this is going to govern gasping. So if that time where if you hold your breath, the rise in CO2 is detected and forces you to gasp for air, that is governed by this apnoistic areas. Again, both areas in the pons. The midbrain. Now remember, the midbrain is located superior to the medulla oblongata and extends from the pons to the diencephalon. Now this picture gives us a nice look at some of the structures of the midbrain. So let's start with the tectum. Now the tectum includes something called the corpora quadrigemina, so this body of twins, two twins, so four bodies that include the superior and inferior colliculi. Now you say, well, what are these for? These are startle reflex centers. When you hear a sound or you see something, your head juts to that sound and your eyes move quickly to that sound. It's a protective mechanism against predation. When we do the sheep's brain dissection in lab, we'll really get to see these two bodies. Next, I want you to look at our anchors, our feet, our cerebral peduncles that kind of anchor and hold everything in place. We have three portions of these peduncles that gives us the superior feet, the middle feet, and the inferior feet. So again, we'll name them the superior cerebral peduncles, the middle cerebral peduncles, and the inferior cerebral peduncles. Now again, let's look at some of our cranial nerves that we just outlined. We have the trochlear nerve here, the facial, vestibular cochlear, glossopharyngeal, that famous vagus nerve, cranial nerve 10, accessory nerve, and then now, remember when we talked about our cervical spinal nerves back in our spinal nerve lecture? Well, here it is, CSN 1, where the spinal cord extends down through those vertebrae. Now, a couple other things to look at. Look at these tracks to those nuclei, the cuneate nuclei, the gracilis nuclei. Look at these tracks. These are the ones we just outlined, right? So you can see those tracks moving up from the spinal cord up into the brain. So now that we looked at the parts, let's see the functions of the midbrain. So it's going to move motor impulses from the cerebrum to the cerebellum and the spinal cord. It sends sensory impulses from the spinal cord to the thalamus and regulates auditory and visual reflexes. Remember those corpora quadrigemina? Well, here they are. We have again our nuclei of cranial nerves, so the origin for these cranial nerves 3, 4, oculomotor and trochlear nerve. We have our cerebral peduncles, the anterior ones that connect the cerebellum to the midbrain, again, anchors. Our corpora quadrigemina, superior and inferior colliculi, consisting of those pairs of twins that are going to help give us those visual and auditory reflexes. We have our substantia nigra, another set of nuclei with the red nuclei. Again, that substantia nigra is going to work in conjunction with the havenular nuclei governing reward addiction. It's a major relay center for neurotransmitters to the thalamus. So again, reward addiction and movement. And the red nuclei, termed red because of its color, it has a high concentration of iron, which gives it that red color, which is important for posture and acts as a major relay center for our brain. Let's take a look at a cross-section, transverse section of the midbrain. And again, we can see our reticular formation. This is part of our reticular activating system that keeps us awake throughout the day. We have our tracts, our medial lemniscus right there. We have our red nuclei and our substantia nigra. Again, substantia nigra is termed this because of its color. It is very dark. It has a high amount of dopaminergic receptors that are receptive again to dopamine. We have a couple other regions, a cortical spinal region, a cortical pontine, and a cortical bulbar axons. Again, that word spinal indicates that these are motor tracts. And lastly, our oculomotor nerve. This is cranial nerve number 3 that's responsible for eye movement. We're going to look a little more into our reticular formation. It serves multiple functions with the reticular activating system, so it helps regulate muscle tone. It alerts the cortex of the cerebrum to incoming sensory signals and is responsible for maintaining consciousness and awakening from sleep. So what are those visual cues? Again, light, the rising of the sun, kicks off this system to keep us awake, whereas the setting of the sun kicks in the pineal gland to secrete melatonin to allow us to go to sleep. Table 14.2 summarizes everything we've talked about, so the functions of the principal parts of the brain. So I like that it colorizes the specific regions and gives you an outline of what we've talked about. So it puts it condensed into a nice, neat little table. This is a good way to make some note cards for each part, for what it contains, what its function is, and through what avenue. So you should break apart this table if you didn't take really good notes on the last few slides that we talked about, because this kind of puts it all together for you. The next part of the brain we're going to look at is the cerebellum. So the cerebellum is going to occupy the inferior and posterior aspects of the cranial cavity and consists of two hemispheres and a central vermis. So we can see that vermis right here. We have our two hemispheres on this side and this side, separated by two lobes, an anterior lobe and a posterior lobe. We'll look at a cross-sectional cut to work through some parts of this brain. So the cerebellum functions in the coordination of skeletal muscle contractions, as well as the maintenance of normal muscle tone, balance, and posture. So we have a lot of Purkinje cells here. And if you think about alcohol and what it does to our body, it affects the cerebellum. So if you think about drinking alcohol in excess and what happens physiologically to you, and how you lose balance, you become disoriented, that's because we're inhibiting the action of this part of our brain, specifically those Purkinje cells. That's why we lose balance and we lose the ability to maintain proper posture. So this is an important clinical target when we talk about alcohol and the effects on the brain. So again, we have the cerebellum here, but a couple things I want you to point out when we put it all together. We have the pons, the medulla oblongata, and then look, here's our cerebral aqueduct, again connecting the third, which would be here, and that 10th of the fourth ventricle. And then we keep going down into the central canal of the spinal cord. So we have those anterior and posterior lobes of the cerebellum. They're subconscious skeletal muscle movement. Now, let's look at these lobes. The flocculonodular lobe deals with equilibrium and balance. And we have the cerebral peduncles again, connecting the cerebellum with the brainstem, inferior to the medulla, middle to pons, and superior to the midbrain. The next portion we're going to focus on is the diencephalon, a super important part of our brain. Well, the diencephalon is composed of the thalamus, so we see the thalamus here. It is about the size of a small egg. We have two small eggs for both each left and right hemisphere. It's tied in by a interthalamic adhesion mass. The hypothalamus, again, hypo meaning below. This is going to be a major part of our brain. This is going to be known as the master of all masters. The thalamus is essentially a relay center. It's relaying signals to the cerebral cortex. The hypothalamus is going to be known as, and we'll draw a line down here, the master of all masters. And that's a pretty strong statement. And we get into AMP2 and we talk about the endocrine system and what this means. This is going to be governing the release of pretty much the entire endocrine system and hormonal secretion through the body, maintaining homeostasis, and running that entire movement of hormones through our circulatory system. Now, the epithalamus is continuous with the pineal gland, which will govern our sleep cycles and release of melatonin, again, hormones. Here's a closer look at the thalamus. Again, look at the size and shape, two small eggs. Here's that interthalamic adhesion mass connecting the left and right portions. Now, again, it's located superior to the midbrain and makes up the walls of the third ventricle. So in here, in that space, would be our third ventricle, right here. Okay. The thalamus contains nuclei that serve as, here's our big word, function, relay centers for all sensory impulses. So when we talk about sensory information coming in via a first order neuron to second order neuron, that second order synapse where those cell bodies are located are going to be right here in the thalamus, and then moves third order up to the cerebral cortex for interpretation. Now we have the relay center for all sensory impulses except for smell, olfaction, right? So that's the only one that will not move through this relay center. And again, it's sending those sensory impulses to the cerebral cortex for interpretation, and then motor tracts will descend their way down. Now, there are a lot of nuclei here that we aren't really going to focus into, like the reticular, the ventral and anterior, the lateral dorsal, all these different portions, the lateral geniculate. We're not the pulvinar. We're not really going to get into specifically what all these nuclei do. Don't really worry. We're just broad stroking the structure and looking at some of their functions. The next portion is the hypothalamus. So we hinted at this a few seconds ago. So it's found inferior to the thalamus, again, hypo meaning below. It makes up the floor of the third ventricle. It has 4 major regions. This is going to control many body activities. So look at pages 490 to 491 to really identify what those are. We're not going to go into too much detail now, but when we do endocrine system in AP2, we will cover this in much more detail. And it is, again, the master of all masters and major regulators of homeostasis. Let's take a little look at some of the regions. So if we look at our key down here, the red is the mammillary region, which is responsible for recollective memory. Now, we go back to memory information begins within the hippocampus and then really gets stored here for recollection, so remembering those memories. Again, this is what these regions are what this mammillary region is going to be responsible for. The next portion is the tuberal region. And again, just to broad stroke it, just go through some general portion. The tuberal region is responsible for emotion, things like rage, appetite, body weight, insulin regulation. It also manages control of the anterior pituitary for feeding. The third region is the supraoptic region. It's responsible for things like fluid balance, milk letdown, and parturition. The fourth and final region is the preoptic region, and this is going to be responsible for thermal regulation as well as sexual behavior. Last but not least, we have the epithalamus. Lies superior and posterior to the thalamus and is continuous with the pineal gland, which secretes melatonin, as well as the habenular nuclei, which are involved in olfaction. One of the last things we're going to talk about are the CVOs, the circumventricular organs of the diencephalon. They are part of the diencephalon. They function to monitor chemical changes in the blood, and this is because they lack a blood-brain barrier. The circumventricular organs are also part of the hypothalamus, the pineal gland, and the pituitary gland. CVOs are going to help coordinate homeostatic activities of the endocrine and the nervous system. So basically, to summarize these circumventricular organs is to say that they are pretty much structures that permit polypeptide hypothalamic hormones to leave the brain without bothering or disrupting that blood-brain barrier. It's also going to allow for substances that do not cross the blood-brain barrier to change. So there's going to help it trigger changes in brain function. The last slide in this lecture is table 14.2. It's a continuation of that table we looked at before. And again, it's a nice little table to lump everything together in one nice, neat little package, giving you the who, the what, the where, the when, the why, the how of each of these structures. This the final part of this table is the diencephalon, the thalamus, the hypothalamus, and the epithalamus, what it's made up of, what it does, etc. So I like this table.